Howdy, junior microbiologists, and welcome to Innate Immunity 3, an evolutionary arms race. So this is the second video dealing with the second line of defense. Chapter 15 in the Bellman text. So here's our flow chart that we've been using to study the immune system. So you can break the immune system up into these two major classes. The innate immune system, or innate defenses, also called nonspecific defenses, and the adaptive immune system, or adaptive defenses, and this is the specific immune system. So this video we're going to talk about the second line of defense. We'll talk about the internal defenses, including phagocytes. So if you've listened to my other video about the second line of defense, then you'll know that phagocytes would include macrophages and neutrophils and eosinophils, and they basically eat the microbes that invade your body. And then these guys, natural born killer cells, they're kind of cool. They do immune surveillance, and they're always looking throughout your body for cells that look weird, for example, cancer cells, and then they stop cancer before it begins. Another second line of defense is inflammation. So usually it's like, oh, I have to take anti-inflammatory drugs, inflammation sucks. But really, it's there to help protect you against microbes. Antimicrobial proteins are also involved in the second line of defense. Those would be considered chemical barriers, as opposed to the external or physical barriers in the first line of defense. And then fever is also a part of the second line of defense, because microbes don't like to reproduce or replicate when it's hot. So let's take a look at the complement system in a little more detail. We've mentioned it, but now we'll look at it a little more closely. The complement system, if you think about what it does. So there's a whole bunch of biochemistry here, but if you think about the end point of complement, and that is to get these complement proteins, C5b, C6, C7, C8, and C9, to form a hole or punch a hole in the membrane, the phospholipid bilayer, here's phospholipid bilayer, of a target cell, for example, a bacterial cell. So once you activate this complement system, these complement proteins, these purple things, will line up like slats in a barrel, make a hole in the bacterial cell, and then the bacterial cell will lyse and die. So way to go, complement. There's different ways to activate the complement system so that you can kill these microbes. There's the classical pathway, and this is activated by antibodies, and we'll talk about that when we do the second series of lectures on the immune system. So the adaptive immune system, we'll talk about antibodies and B cells. There's the lectin pathway to activate complement, and we talked about that. We had a cartoon last video. I think these molecules called lectins can bind to proteins on a bacteria, and then, well, actually it says here to specific sugars on a bacteria, and then macrophages can recognize the bacteria and kill them. And then there's this alternative pathway to activate the complement system, and it's kind of activated spontaneously. So let's take a look at our check marks over here. So complement system, it's a set of plasma proteins that result in killing of foreign cells. So complement proteins get together, kill foreign cells like a bacterial cell. And they call this this activation of the complement proteins, they call it a cascade, because one enzyme will activate the next complement protein down the line. And then that protein will be an enzyme to activate the next complement protein. And the end product of the cascade is this membrane attack complex, this purple thing here, that forms this hole in the pathogen's plasma membrane, and then the contents will leak out and the bacteria will die. Complement proteins can also act as opsonins to opsonize microbes. So when antibodies are coating the target cell along with activation of complement, that's called opsonization. And then complement proteins can also stimulate white blood cells, and white blood cells come in and can destroy the pathogen. Complement proteins can be chemotactic factors, and then complement proteins can indirectly trigger inflammation and fever, which can also help kill microbes. So a little bit more on opsonization. That's just when you're coating a microbe. So opsonization can occur when this protein, C3b, actually coats the bacteria, and then that creates attachment points for white blood cells, such as macrophages, which can then attach to the opsonins in the microbe, and then the macrophage can do phagocytosis or eat the microbe. And then we see here C3a and C5a act as chemotactic factors, and that just, a chemotactic factor is just a molecule that shows white blood cells where to go to get to the bacteria. And then C5b through C9, so C5b through C9 forms the membrane attack complex that we just talked about in the last slide to kill bacteria. Okay, so that was second line of defense, complement proteins. Now this is more on the second line of defense, inflammation. So we've gone over this cartoon previously. You have the splinter go in, these green things are bacteria. Damaged tissue releases inflammatory chemicals, and then neutrophils come to the area where the bacteria are and do phagocytosis. But in this cartoon, we're going to talk about how this cell right here, it's called a mast cell, sitting just inside the wall of the blood vessel. A mast cell can initiate inflammation by releasing inflammatory chemicals, and inflammation results in dilation and increased permeability of blood vessels. So here's blood vessel down here. So dilation just means the blood vessel gets larger in diameter, so it dilates. That means more blood can come through, more blood carrying white blood cells. And then increased permeability, so the attachments between these endothelial cells becomes leaky. Vasoactive chemicals from mast cells and also basophils inside the blood release these vasoactive chemicals, and the chemicals include histamine, prostaglandins, bradykinin, leukotrienes, but the granules inside the mast cells and basophils degranulate, and then they release these vasoactive chemicals. And vasoactive, vaso means vessel, and active means active. So these chemicals act on the blood vessels, vasoactive. And then there's a cytokine called a chemokine that recruits the leukocytes to the site of infection. So here's the site of infection. So some of these inflammatory chemicals would be chemokines, and the neutrophil would move along that chemokine gradient to get to the battlefield and destroy all the bacteria. Migration, inflammation involves migration of phagocytes, so that just means the phagocytes emigrate out of the blood vessel, and then they migrate along this chemotactic gradient till they find the bacteria. Inflammation also results in tissue repair, so that's kind of cool. Inflammation is a complicated process, but these are the highlights of inflammation. Signs and symptoms of inflammation, you probably already know this because everyone's had inflammation in their lives, are redness. So if you have light colored skin, you can see the redness in inflammation. You can feel that it's hot. If something's really inflamed, you can feel it's hot. It swells, that's edema. There's pain involved in inflammation. And then loss of function. If the inflammation is great enough and the pain is great enough, you can't use that body part. Okay, so let's look a little more closely at this second line, inflammation with dilation and increased permeability of blood vessels. So your body's initial response to injury or invasions, such as bacterial invasion here of pathogens, is very localized. So you don't get an inflammatory response over your entire body when you get a splinter in your finger. It's very localized. And the dilation and permeability of the blood vessels are likewise localized. You don't want all your blood vessels to dilate at once. You'd have really low blood pressure. And then we talked about some of the chemical mediators involved in dilation and increased permeability of blood vessels. And then these are the cells, the basophils, platelets, mast cells. They release chemicals like histamine when they're exposed to complement fragments. So if there are some complement fragments in the blood vessels here, they would, the mast cells and the basophils would recognize those complement proteins, and then in turn the basophils would release, for example, histamine. So second line, inflammation, diapedesis, and chemotaxis, and I should say margination as well. And we talked about this in the last video. These inflammatory chemicals will make these endothelial cells sticky. Neutrophils, in this case, in this cartoon, will stick to the endothelial cells. That's called margination. So you get the white blood cells to accumulate along the blood vessel wall. And then diapedesis is when they emigrate, the white blood cells emigrate, or diapedese, so kind of crawl or squeeze through the lining of the capillaries where these endothelial cells become loose. And then chemotaxis, the neutrophils do chemotaxis. That's the movement of these neutrophils toward the chemical stimulus. And the chemical stimulus is this gradient of chemokines, which are generally called inflammatory chemicals in this cartoon. So here is a cartoon of a capillary. Maybe you remember this from Bio 202 when they talked about blood vessels, but here's a blood flow going in through an arteriole. And then this is before, so blood moves into the capillaries and then out the venule. And then after you dilate your arterioles and capillaries, so here's a mediator like histamine telling these endothelial cells, or the smooth muscle cells, sorry, around the blood vessels to relax. And then the smooth muscle cells relax and the capillaries and arterioles dilate or get bigger, and you have increased blood flow through the capillary. But you also, this increased blood flow in the capillaries causes reddening because you've got more volume of blood, red blood flowing through these arterioles and capillaries, so you can kind of see that through light skin. And then heat, because you've got this rush of blood now with these dilated arterioles, and that feels warm to the touch. So this increased blood flow, that's BF, delivers monocytes and neutrophils. In this cartoon, it shows a neutrophil to the battlefield, to the site. The first white blood cells to arrive at this infection site are neutrophils, and then the monocytes come. Remember, monocytes are in the blood, and then when the monocytes get into the tissue, they're called macrophages. And we've talked about this, the phagocytes roll along the blood vessel wall to get close to the damage. So if you go up here where there are no inflammatory chemicals, these endothelial cells won't be sticky in the blood vessels, and neutrophils will not marginate up here in the blood vessel. They'll only marginate where these inflammatory chemicals have told the endothelial cells to get sticky. So this is the cartoon of the normal permeability of a venule. So that's upstream of a vein. So here's the venular wall. These are endothelial cells. Here's a monocyte inside the blood vessel. Here's some red blood cells. When you increase the permeability of a venule, you can see, look at the diameter. So that in a normal permeability, well, the normal diameter is here, and then you can see the diameter of the blood vessel gets a lot bigger in inflammation. And also, look at all the leaks between these endothelial cells. And then the endpoint of inflammation is tissue repair. So here's the tissue, got some cut, had some inflammatory processes, and then over time, the cut heals. That's tissue repair. That's at the end point of inflammation. Okay, the second line of defense includes getting a fever. So this is kind of cool. So fever results when chemicals called pyrogens are released. So have you heard of a pyromaniac? So they love fire. So a pyrogen is genesis is creation of heat, pyro. So fever, you get a fever when pyrogens are released. Okay, so check it out. Here's your wound. So you got a wound, and chemicals like pyrogens were released at the wound site, and they were released at the wound site by phagocytes like macrophages and neutrophils, and then those chemicals got in the blood vessels and traveled up here to the hypothalamus. And those chemicals talked to cells in the hypothalamus and told them to secrete prostaglandins, and prostaglandins will reset the hypothalamic thermostat. So what's your thermostat usually set at? What is it, 98.6? Well, they'll reset it. These prostaglandins will reset that thermostat to a higher degree, so that, and then your body temperature rises, you have a fever. And then you have some behaviors like shivering, but you also have when you have fever, you have a higher metabolic rate, your sweating is inhibited, and you have generalized vasoconstriction of your blood vessels. All these increase your body temperature to the set point set by the hypothalamic thermostat, which the new set point could be 103 degrees. So there are these molecules called endogenous pyrogens, and they include the interleukins like IL-1 that are released from the macrophages here at the wound site, and those are endogenous pyrogens. And then you have exogenous pyrogens, including LPS. And if you remember, in your gram-negative cells, in that outer membrane, you have LPS, which is actually a bacterial toxin. So an exogenous pyrogen like LPS can be a bacterial toxin. It could be exogenous pyrogen, could be the cytoplasmic content of bacteria. It could be antigen-antibody complexes. And then also, we talked about this already, endogenous pyrogens are released by phagocytes here at the wound site. We just talked about this, the exogenous toxic pyrogen, LPS. I told you it's found only on gram-negative bacteria. It can stimulate fever in the host, that's you, if you have a gram-negative bacterial infection. And then that LPS, the lipo part is the toxic component of LPS. And this is just a little tricky word stuff. Pyrogens can also be called pyrogenic molecules. Don't confuse that with pyogenic, which means the creation or genesis of pus. Might be it. That might be it. Nope, nope. As your infection gets under control, the pyrogen concentration goes down and your thermostat is reset to your normal body temperature. If your fever gets too high, your proteins will denature, nerve impulses are inhibited, can lead to hallucinations, coma, and death. But the increased temperature that's not too high enhances the effects of interferons. Remember, those are molecules that can warn cells of a viral infection. Increased temperature when you have a fever inhibits the growth of some microbes. And that increased temperature, some scientists think it enhances the performance of phagocytes, so they eat more readily. And the increased temperature also helps in the process of tissue repair, which is the endpoint of inflammation.